I welcome all the participants in the Science Dialogue series. As most of you are aware of about this endeavor that we are organizing regularly this Science Dialogue series free of cost, no charges, and even we don't offer any uh, honorarium to a speaker. So I'm thankful to all the speakers who are uh, delivering lecture in the Science Dialogue series free of cost. And in this series, you all are aware of about today's lecture uh, on yeast expression system to produce recombinant protein using yeast. And today's speaker is Dr. Bijesh. He is my friend also. We have spent 15 days at IICT Hyderabad for technology-led entrepreneurship course. He did his PhD from CFTRI, Food Technology, Mysore. And then he moved to Texas for postdoc fellowship. And nowadays he's scientist at IICB Kolkata. As you know, as a researcher, he has also published several research paper, review paper, delivered lecture. So I welcome Dr. Bijesh in the Science Dialogue series. And participants are from all over country, sometimes outside also. So thank you for accepting our invitation. Now platform is yours.
Okay.
If possible, just put light on your face.
Yeah,
Sometimes
but what happened is light is coming from my
back.
backside, so
So don't worry, don't
that's why
worry.
my face is not so clear. So, but it's
Uh,
fine. We are more focused on slides and
like, yes, yes,
content, so it should be fine. So
yes,
now
yes,
what I'll do, I'll put my
yes.
full
So open your
screen
slide now, huh? Yes, yes, it's
slide.
visible.
Okay, then it's okay. So, so thank
Very
you, Gopal, and whoever is behind, because it is should be a team behind the Science Dialogue series. So there will be some more people, I think, like involved.
mostly my student. My student is
Huh.
helping me in preparing all these flyers and all these things, Anna.
Yeah, that's right. So it's a effort actually from a lot of people. So thank you for everyone for the invitation. So as he mentioned, I'm a scientist at CSIR-Indian Institute of Chemical Biology, Kolkata, in Cell Biology and Physiology Division. So you can contact me. I have given my email ID on the first slide itself if you have some query. So this talk is more like an interaction discussion. So if some query is there, you can ask or you can send an email. So, so it's more like we have a research problem to solve. So what we will do? So we have a research problem that is like functional analysis of a human multi-domain folate receptor enzyme. So this is like a hypothesis. I'm just putting for like hypothetical situation. So you have like a multi-domain protein and you want to work on that one. So we will do like a kind of practical approach. So this will be useful. Like we will be learning step by step how we will solve this situation. So you can thinking about how you will do this. Like you have a multi-domain protein, how you will do the functional analysis. So one easy way is like express it in like some system like E. coli or yeast. So what mammalian cell or cell-free system. So now we'll just look at bacterial system means it is not so suitable for like a multi-domain and complex protein. Yeast, insect, and mammalian cells are pretty suitable for this purpose. A cell-free system people generally don't use for normal proteins. Cell-free system is people generally use for toxic protein that are harmful for the cells. So if the protein is produced, that cell will die, like yeast or bacteria, insect cell, mammalian cell. So possibility for them to die is very high. So those kind of scenario, people go for cell-free system. So why we are not using bacteria or insect or mammalian cell is mainly because as you can see, bacteria is there are like lot of advantage. Yeast, baculovirus or insects and mammalian. Okay, so if we compare, one main thing is like bacteria is not so
You
suitable
can
for.
hide this line, bottom line presentation.
Oh,
Just
okay.
click on that hide.
Yeah, one second. Okay.
Yes, thank you.
So you can see the bacterial system, it's not so suitable for complex protein because it's not good for protein folding or handling glycosylation or any kind of post-translation modification that is usually seen in eukaryotic proteins. So when we come to eukaryotic system, we have these three: yeast, insect, or mammalian. But you can see the problem with the yeast is it is not doing everything. Every post-translational modification is not so robust. But main benefit is in cost. So compared to the other two eukaryotic systems, it's pretty cheaper. And complexity, like we can do in like minimum, like we don't have to worry too much about the contamination during culturing and all compared to other systems. So this system is little bit like bacterial system. It is simple, easy to use, but there are like little bit difficulty assigned because the growth is not so high. As you can see, it's 30 minutes for bacteria, it's 90 minutes for yeast. And yeast, another is like the presence of cell wall make it little bit difficult to work with the yeast system. So bacteria is cell wall is, there is no cell wall. Yeast, but it got like a thick, very thick lignocellulosic cell wall. So transformation efficiency is very less, and getting the protein out from the system and working on that protein is very difficult. Plasmid isolation from yeast, generally many people won't do it, that itself. So these are the some of the problem with the yeast. But when it comes to cost, it become like compared to bacterial, a mammalian or insect, that system is very cheap. So when we have any eukaryotic proteins, always better to start with the yeast system. So that's what we are also going to do because we have multi-domain protein. So now you have the protein which you want to express in a system or yeast system. So first thing what you want to do is go and see the sequence and whatever information available about that protein or gene in like something like NIH, NCBI database. So if you go and see there, there will be like some sequence related information. So there will be a lot of information. As I highlighted, our protein is undergoing glycosylation. So that is one reason why we selected yeast, because yeast can do this post-translation modification like glycosylation. And another reason, And another reason is the structural complexity. As I told earlier, multi-domain proteins are not so suitable to express in bacterial systems. So, and if you have like too many multiple like some disulfide bonds, those kind of our protein is having, whereas especially folate receptor domain in human, that is not have like kind of 8 disulfide bonds. It is possible for bacteria to make these bonds, but what happens is like when it comes functionality, what happens, they don't fold properly, or we have to go for like some kind of bacterial systems like origami systems or something to get little bit better. But for structural studies it may be a bit suitable, but for functional studies it's better not to do in bacterial system, especially multi-domain comes into picture. Go for at least basic, we should start with the yeast system. So now we have our protein bit complex and it is undergoing glycosylation. We know a little bit about this one, our this NCBA or sequence structural information. Now we have mainly two type of yeast system like Saccharomyces cerevisiae and Pichia pastoris. Earlier name was Pichia pastoris. Now people are using this Commegatella puffy, and we will use Pichia pastoris because it's convenient, but taxonomy is a bit changed now. The people understood actually that Pichia is a bit different, and that is another one only. So whatever commercial or majority what commercial people use is actually the not the Pichia pastoris that was meant to be, and it is and Commegatella puffy. So we are using Commegatella puffy for names. We just address it as Pichia. So as I mentioned earlier, we have two yeast system, and we have a protein from human. So that is human multi-domain protein we want to work. So next question comes about the codon optimization and codon usage bias. So we have a human protein we want to express in yeast. Problem means like yeast got its own what you call preference for codons. So you can see like leucine, you have UA, UUG, CUU. So there are like lot of codons, triplet codon that can code for this amino acid. So it is same for like most of the amino acids. We have multiple codons. So each organism have a preference for some codons. So if you take the sequence as such from human and try to put it in yeast, the protein won't express so well because yeast won't have that much preference for this that codon, but human is we are having. So first thing what we had to do means we had to take the sequence, put it in like some kind of lot of softwares are available online for codon optimization, and paste it there, give your preferred organism, whatever. So it will automatically change the sequence according to this codon preference. So that is the first step we are also doing here. Okay, so now we are actually doing our sequence changing to make it suitable for our yeast system. Now the question comes which vector, which plasmid we will use. So now we know our protein. So what we want to do, we want improved expression of the protein. We want enhanced solubility of folding. We want to detect the protein after it is
So, what are
expressed.
the differences in codon preference in yeast and the human?
I didn't go exactly through that one, but that's why I told like a lot of softwares
Generally,
are
in case
available.
generally in case of that bacteria,
Huh.
we have that data that some codons are rare codons. That's why Rosetta Stone is used to overcome the effect of codon biasness, nullify the effect, because we add the extra tRNA gene which codes for rare codon. So that Rosetta Stone can compensate the codon biasness. No need to change the gene
Huh.
sequence.
But here in this case, what happens is like we want better, as I told, it will code. It's not about coding. It will code the protein, but the yield is a problem. You won't get that much yield with that one because there is a bias
That's
for
why,
that codon.
yes. So means
So
software will
there
take
are
care
some
of all these
difference.
things.
Yeah, but there are like online tools. You are taking a lot of classes on that one, and so like, tools are there. So it is very pretty easy actually. That is why I'm not going to. I actually didn't prepare too much into
Yes,
deep
yes,
about
yes.
that codon,
Please
but
go
there
ahead.
is like lot of bias with each organism. So
Yes.
other either we have to do or we give our sequence to the company for synthesis of gene, and we give the name of the organism what we want. They immediately convert that one to
Okay.
that organism whatever preferred. Then only they will synthesize our gene. So we are also doing the same thing. So we
Okay.
will give to the company, and they have that their own software each one, like GenScript or
Free software
any
available
Eurofins.
for this.
A free software is available.
Okay. Okay. Good.
So we can check before giving that one, like we can compare also,
Okay.
like whether our sequence is matching with whatever the company is providing or not.
Okay.
It's possible.
Please continue.
So now we want to use the vector. So as I told, we want improved expression, enhanced solubility, easy detection, simple purification, and some kind of protection from degradation. So now we want to select a vector or plasmid where we want to insert our gene for expression. See, improved expression generally we can have like a very robust promoter, and that promoter drives the expression. When it comes to solubility, our protein I told already, it's like a multi-domain folate receptor protein. So it's a cytoplasmic protein. That's kind of information, as I told, we will get from online NCBA database and everything, or we can go for prediction also. So if it is like a membrane protein and all, or it's like kind of fat soluble kind of protein, so the normal expression will be difficult. So that time we can actually go for kind of increasing solubility, folding, those kind of domains we can insert to our sequence. Dictation again, we have to have tag. Either we have to have some method for dictation our own protein, like antibody should be available. Otherwise, we add one extra tag like a hashtag or something for easy detection. Purification also, this same kind of tags or kind of different tags we can use so that it can be purified. And protect from degradation, we can go for multiple approaches like don't go for secretory sequence so that it gets secret outside the cell. And we have some kind of protease inhibitor like in the system so that it can protect the protein that is secreted. So many approaches can be done. So what we will do, we will start with some simple system. So first we will see what are the plasmids available. So there are like mainly four types of plasmid available for yeast expression. One is integrating plasmid because it is integrating the genome. Second is centromere. It acts like a plasmid because it is having a centromere. So it divides just like chromosome of yeast. Yeast replicating plasmid is generally not for protein expression. Yeast episomal plasmid is again not integrating genome, it stays outside and does expression. So we are interested in two different approaches that is meant for protein expression. So these two are yeast integrating and yeast episomal. So I'll just show you what is these two. So as you can, episomal means it is not integrating genome, it stays outside. The benefit is you can see like you can have multiple plasmids. So the copy number can go to like 100 or 200. So based on plasmid, you will have many plasmid. So you have multiple copies of a gene. So you have a robust expression of your protein. But there are some disadvantages because how they used to retain the plasmids because it don't need the plasmid as such. Okay, so what will happen is like if you don't give some kind of selection pressure, it will shed the plasmid. So we select the yeast and remove the selection pressure because some antibiotic resistance or something. So there is no need for the yeast to retain the plasmid. Then what will happen? That plasmid will start shedding the plasmid and it won't have the plasmid and our gene. Then there is no protein expression. So long term that is not good. So what we had to do means we had to integrate it to the genome. So that is the second approach. So, but the problem again, you can see there is like only one copy number, or like very rare cases, it can go little bit higher copy number. That's it. So the expression is coming from only one gene. Then yield will be kind of less. Only benefit is like you can go for like scaling up and all. That is easy. So industrial scale production generally we have to go for integrated expression because it is pretty cheaper. You don't have to give any kind of selection pressure. You have only like a basal media and some inducing agent. So that's it. It works. We will see all these things in detail. What is episomal and integrative expression? So we have a protein with us. So the first step what we will also do means we'll go for an episomal expression. Why means we know the protein yield will be there and we are not going for like large scale production anyway. So that is what most of the people will do. First step is go for episomal expression so that we will get good amount of protein to work with. So now we will start our work. So we have the gene, we got a plasmid. I'll go to the plasmid again. So we got our gene synthesized and either the company will put into that whatever plasmid we are telling. Otherwise, we can do like recombination means ligation and get it done and put it in a yeast bacterial system initially so that it can amplify because we have only very little plasmid synthesized and got it from the company. So one second. Okay, so next step is like we have to go for yeast transformation. So compared to bacterial transformation, yeast transformation, what happens, I told it's a little bit thicker cell one, so the approach is little bit different, but still it is kind of similar. We can go for electroporation, we can go for chemical based or lithium acetate based transformation. So it's not so difficult. Then next comes selection and verification. So we'll go by step how whether our protein is expressed and whether we can detect it properly and whether it is functional. So this one we have to do. So first is we'll go for, as I told, we'll go for an episomal expression so that we know we'll have a little bit yield of our protein. So this is the vector we are selecting for our work. It is known as PS to NTA. It's commercially available vector. So that is available from Thermo Scientific and I think like Adgin also may be giving. And there are like PS2NT-B, PS2NTC, many are there. Those three minima are there in that category. So there will be like little bit difference among them in this construct, whatever is there. So we will explain what is there in this vector that is useful for us. The first one I have highlighted. This is the region, the red circle. This is the region where our gene is going to get inserted. Now our gene is not there. So it is having one GAL1 promoter. So that is like inducible promoter. So we give galactose. So what will happen? This promoter get activated and drive the expression. And it is having a T7 here promoter sequence that is useful for sequencing. Actually, it is not actual promoter; it's a promoter sequence. So when we insert our gene and we want to confirm our gene is there or not, so we can send to company and tell them use T7 promoter sequence forward. Usually it will have a reverse also, T7 reverse also. So what they will do, they will have this one. So these are universal, what you call sequencing primers. So we don't have to give these things. So they will do and give us back. So it is having like a start codon. Then it is having histag at either end. So C terminal and N terminal, two histags are there. You can see one is here and one is here. And it is having one express TM tag that is also same kind. We can go for detection. It is having also like express TM antibodies are there, so we can use. And histag also it is same thing. We can also have antibodies, histag, and we can use nickel and TA column for purification. Then our multiple cloning site comes. Again, it is having V5 tag that is also useful for all this purpose. Again, one more histag, and we have terminator. So as you can see,
I don't understand why there are two promoters. Only purpose is to use it for
that is
sequencing.
yeah.
T7
T7 promoter is not a full sequence. It is just a very small sequence, just maybe like 20
promoter is
25 base.
promoter
That's
is
it.
all T7 promoter size is already of 17 nucleotide long.
Yeah, that's what that is I told. This is the bigger one is actual promoter. That is GAL1
Okay.
promoter. This one
Mhm.
is only for sequencing, but it
So
is having
that
only few bases.
gal one part can be used as a sequencing. What is the problem?
We can use. The thing is like these companies won't have GAL1
Okay.
promoter because it is not so common. But
Okay.
T7 what happen is like it is only used for sequencing. So they have the sequencing, they will have all these primers. We don't have to give them. So it is like a kind of many like universal for like many vectors to put one T7 forward and reverse for easy to sequence actually. So we don't give this T7 primer our own. We just have to give vector and
Okay.
tell the sequencing company. So there are like universal sequencing primers. So it is included in that list. So that is why they are giving that T7 promoter sequence at the beginning and end also. They will usually they will have these to end. So
Okay.
whatever we do, we have to confirm after sequencing like whether gene inserted or not. So that is in frame, that is in proper, there is no sequence errors or mistakes. For that purpose that is easy actually. So that is why they are giving. But
Okay.
as you told, we can give our own gal1 promoter primer, but they won't be having that one. So we have to what you call synthesize it and aliquot it and send to them. And that's bit headache actually. So this become, so I think for
Okay.
convenience purpose, this one is good. And there is a list online available universal sequence primer. So in that one you can see that many are there. Some are not so universal, so it won't be like not available. But I think slowly, when we like a lot of people start working on Galvan, then what will happen? They will make one Galvan promoter sequence also like universal. So we no need for like a T7 promoter then.
Okay, okay,
It is
great.
only for sequence. There is not act like a promoter. It is only like 20 base, 20, 25 base, only for sequencing purpose.
So this is downstream of Galvan promoter or upstream of the Galvan promoter?
It's after downstream of Galvan promoter because that it's
Yes.
not for coding. That is before ATG. It comes. It got its own ATG, so it comes. So now what happens is like while explain what we want in our gene. So as I told already, it is having histag at either end.
Okay.
So we don't want all this histag and everything to come. Why it is there means sometimes N-terminal histag will affect our protein expression or
Yes, yes,
protein
yes. So
activity.
we add
Sometimes
in normal
C-terminal.
bacterial system also we add at both terminal, है ना? Tag. Yes,
So in that case, actually, we can select with what we want. We want
yes.
C terminal or N terminal. Accordingly, we can actually go for that tags. But they are just giving two tags at either end of cloning site. So one express and V5 is also there. So we can opt actually what we want, and then we can put like stop codon also. It is having a stop codon after this six his, but if we want only this MCS and we don't want V5 6s, after MCS put one stop codon to our gene, so we will have only that the initial histag and SPSTM tag. SC terminals. Now we will come to the vector. What are the actual characteristics of like yeast plasmid is next of rest of the things. The first origin of replication is for bacterial host. So this promoter, this particular one is having one F1 ori. So that is not needed in our case. We are going for like a normal expression. So this one is active, become active only in, I have written here, when we use like helper phage. So under that category, it become activated and it start working. But for replication yeast, it is having 2 micron ori. So there is an F1 ori that we don't want, and it is not functional in our case. In our case, there is 2 micron ori. This 2 micron origin of replication or 2 micron ori that actually help the plasmid to replicate in yeast, our yeast. The first thing we are doing is like Saccharomyces cerevisiae. So this one is suitable for Saccharomyces cerevisiae. This ori, normal ori for bacteria. This ori is for original replication is for yeast. So it is having this. Then another characteristic it is having this ura3, that is the gene name is orotidine 5-phosphate decarboxylase yeast. That is for actually selection. We will discuss more about that yeast selection. It is having ampicillin resistance, that is for bacterial selection. So in Saccharomyces cerevisiae, what people done that antibiotic resistant selection is not so robust. So what people done is like they deleted some essential genes. So like one is like uracil production gene. Here in our case, ura. So that now what happens is like our yeast won't survive in a medium where uracil is not there. So now we give a plasmid, then what will happen is like it can survive. This ura gene is there. So this gene act as like synthesizer of uracil. So what will happen is like it become easy to select. Only when our plasmid is there, it will grow. So it is like what we call oxotrophic yeast. So it is not having like what you call it needs some nutrient. So it is oxotrophic, and either we have to supply the nutrient for that to grow. That is in before inserting of this one plasmid. After insertion, you remove this complete media. Don't give ura. Then what will happen? Every wild type yeast will die. Only that one is having this plasmid with this ura gene that will only survive. So we'll explain again in detail. We'll go step by step. One second. So, okay. So I explained already. We have the plasmid. This was a multiple cloning region, and we are inserting our gene here. Okay. So I have given this construct picture. So this is the promoter. This one is the insert, and it is actually his tag is there. We are giving like a N-terminal his tag to this one, and rest of the things are there. So this one we can get it synthesized also. Like if we give it to company, they will get it done. Otherwise, we can get our gene and we can insert ourselves with restriction cloning to this region. Then we can verify with the sequencing with 37 primer. So it is fine. So first step is of course bacterial transformation selection. So we have like ampicillin for our plasmid selection, and aldehydic acid for DHP alpha. We can use that one. So we have first step, that is amplification, that we done. Okay. Next step, we are coming to the yeast. So there are three methods generally for transformation, that is lithium acetate, spheroplast method, and electroporation. Saccharomyces lithium acetate was pretty well, so no need of electroporation. But electroporation actually was efficiency is pretty high, so that is also people use. So I used in this case lithium acetate method. So we'll see. Again, I'll just tell about selection. The strain we are using is BY4741. So this strain is also trophic for uracil, not only uracil, it's methionine, histidine, and leucine. So these four are like deleted, or this gene for synthesizing these four are deleted in this uh, yeast. So when we are giving a medium, defined medium here, what will happen? There is no uracil in that medium. So there is no synthetic defined medium. Okay. Now we are giving our plasmid. So what will, the plasmid will make that uracil because of ura3 gene. So that plasmid will get retained. Okay. So transformation is done. Next step, what happened? Like our protein is not yet started producing, because our protein is produced only when we are adding galactose, because it is a galactose inducible promoter. So initial phase we have done selection, we will grow yeast to some extent. We have some yeast with us. So this is like representative picture I have given. So this one is like an oxotrophic. I told that urea plates we selected, we grown to some extent. Then we add galactose media. So galactose induce our protein production. So that we can do, we can go for Western blot. Here we have his tag. So go again his tag. So then what will happen? We will get our protein produced. This is like symbol. Now what happens is like, but our approach failed. We want functional protein, but somehow our protein was not functional. So when we checked for functional activity, what we found, the protein is not functional. So we have to see
So
what.
maybe you have shown multiple bands. So maybe your protein getting degraded. Back, go back. Yes,
Yeah,
here
that is
you have
there. That is
multiple bands.
I'll come to. I'll come to.
Okay,
That
okay,
is it.
fine.
I'll come to
Continue.
this one. So degradation is not a big problem actually. What will happen is like when we work with protein, the proteins actually like we won't get every 100 percentage non-degraded protein. Some protein, of course, will undergo degradation because it will have multiple domains. Some domain get cleaved because of some reason. So to some extent we can optimize those one and get that one. But as you told, here the degradation is happening because it is like the last point I have given, that is intracellular protein production. So the protein is produced inside yeast. What will happen is like yeast got very thick cell wall, as I told. So breaking the cell, taking the protein outside, that is a little bit difficult actually. So we are using some crude method. And if we use some enzymes, we are using some enzymes to make uh, to degrade that lignocellulosic membrane. But still, what will happen? The cost will increase. So there are like some protein to get the protein outside also. But coming to the first point, I'll tell one, the protein is not active maybe because of yeast Saccharomyces. I told Saccharomyces is having a tendency to hyperglycosylate protein. So what happens is like it's not good for like those kind of system if we are not getting activity as I told. So pitchia, what will happen is it don't do hyperglycosylation. So now we are thinking about okay, we will move to pitchia system because the glycosylation problem will nullify to some extent. Second is like, okay, we want to work too much with the protein. I told we need to use synthetic selection media just to retain plasmid. So the cost is too high. So we have to have a synthetic defined media and induce with the characters afterwards. There is no way we can get rid of the defined media after transformation. So it is not sustainable for long time, or we want to work with protein and troubleshoot. So it is difficult. But and the third one, of course, it is intracellular. So we thought, okay, we'll move to PGM and it don't hyperglycosylate. There will be like 10 minus units or something. That much only it will go like an average. And no problem. If we go for integrated genome, then next what will happen? It will be inside the gene. So it won't shed. No need to give any synthetic media. We can give our normal media, whatever wild type yeast is growing, and it produce. So it's not a problem. And for pitchiatis, inducing instead of galactose, we can use methanol. So comparatively, methanol is very cheap and scaling up. Like if you have like mass amount of protein, so it become easy actually. So next third is like intracellular protein. As I told, like the protein is getting degraded. Then what will happen is like go for like a secretory signal so that we don't have to break the yeast apart only. So it will secrete the protein. So it become easy. So we are modifying. So we are modifying and we are going with Pitchia. So how we will do this one? So you see the previous plasmid PS2 NTA, it was actually like around 6,000, more than 6,000 base pair. But this one become very small because it is 3,500 93 base pair. So this one what we are using P pi C Z alpha A. So this is the name of the plasmid. There are a lot of plasmids are there. One reason the plasmid is small because it is not episomal plasmid. But it don't have to stay outside. So many things which is needed to stay outside is not there. So it can go and integrate to the genome and express the protein. Now we will see what else are there in the protein, I mean in the plasmid. So this red circle, as you can see, this region contain the multiple cloning site. It is having one AOX1 promoter. That is the promoter that is going to drive other protein expression. And what's the name? I need to mention. So, so this one is methanol inducible promoter. So we use methanol, then the promoter acts and the gene expression starts. I already told we have a secretion signal, alpha factor secretion signal. So that what will happen? It will take the protein outside from the pitchia to outside. So we are solving another problem also. Then our multiple cloning site will come. Then we have two tags, MYC and 6is. So again, we will go with 6is, that is like widely used, and it is having AOX1 terminator. Again, you can see it is having only bacterial origin. There is no another one F1 ori or 2 micron ori or anything because it is integrating, no need of any of these kind of sequence. Then another thing they have done is like they have given only one gene, bleomycin resistant gene. R means it is bleomycin or ceosin. So we can use either of this one to select. And this is applicable for selection in yeast and bacterial system. So we don't have to have separate antibiotic or anything, and we don't have to insert selection pressure because it is integrating. So once we selected and we know this is our strain, we can use any like wild type media. Only like we have to use for inducing protein, we have to have methanol. So methanol inducing promoter, that is the only thing we have to take care. So it becomes cheap, but as I told, it's only have like only kind of one copy number in the organism. So the yield will be like less, but only we can go for scaling up. We can go for higher volume so that we can increase the yield like that. So now this is the promoter, and here after secretion signal, our gene is coming. Here it is, our gene is coming as it is having the secretion signal also. So as I told, we can go for like selection in bacteria with this one and ceosin, and ampicillin we can use for E. coli system. And I'll just tell a little bit about this gene integration. So what happens is like this pichia is having one alcohol oxidase gene, AOX. So we have our gene inside in between this AOX1 promoter region. These sequences are there in between 5 prime and 3 prime alcohol oxidase promoter region. We are having this inside. So what will happen? It will go and bind this one, this wild type AOX1. You can see this wild type pichia, okay, in pichia genome. So when this yeast is replicating, next time what will happen is like this, they will use this as a template also. No? So this template will get incorporated to the next, actually when replicating, what will happen, it become part of the next yeast or baby yeast, whatever you can mention. Okay. So after some time, when we give selection pressure, then what will happen is like our genes will get selected. Okay, so that we have a no, this selection is done. Sorry, selection after insertion is done. It's not of that one. So wild type will slowly get integrated with our gene of interest because of this one is there. Okay, this our gene of interest is there in between. So slowly, our gene get multiplied in that system, Pichia systems. Then what will happen is like we have like what you call few integrated and few non-integrated. Okay, then we go for again like plating, go for one more plating, and go for one more confirmation sequencing, and we have to make sure like our gene of interest is there, pick the colony and go forward. So it is little bit tricky. That is a problem with bacterial and this one. It become like little bit tricky. We have to make sure our gene is getting inserted, and it is not getting inserted in such a way that it is expression is getting less. So it can go wrong here and there a little bit also. There are some problems also, but theoretically this is how we get it done. Okay. Okay. Then we have, okay, theoretically we have our gene get homologous recombination is done. Our gene is getting incorporated there, and slowly it is replacing the other gene. So our gene is there. We can go for induction of the protein with methanol. Okay. So this step, what we will do, we'll go for induction, and we go for a little bit scale up. These are representative pictures. This is not the actual one I have done. I took from Shutterstock. Okay. So before going further forward, actually we can do like it is secreted. So go for like uh, take the secreted one, and you can actually check whether your protein is expressed or not. Then you can go for histag also. Check with the histag whether that protein is in you are this one or not. So here you can see like with which what you have to do, meaning you have to do time course expression also. 6 hour, 24 hour, 30 hour, 48 hour, 72 hour, like that. Because at some time point your yield will be very high. You can see like 48 hour we have like pretty good yield, and 6 hour we are getting like good yield. So sometimes what will happen if you don't have to go for like that much yield. 6 hours should be sufficient to stop after adding methanol. Stop the this one at 6 hour, take the culture, and go for purification of your protein. Now what happens is like we
But
have gone for,
expression is not increasing by increasing the time. Expressions are
yeah, that's what. So 6 hour is pretty good enough in our case actually. No need to go for 48 hour, because 72 hour is actually it's decreasing only. The protein is getting degraded. So
yes.
I feel like
6,
6 hour is good
24,
enough in
30,
our
and 48. Up to 48, it's almost similar
yeah.
expression.
It's a bit stagnant. So that is why our protein is it took some time for us to reach actually, because initially as I told it is really tricky to work with the system, especially pitchy and all. It was little bit tricky,
And you have
but once things
and
get
and this is
adopted,
Western
then
blot.
yeah, this is the download picture.
So why not SDS page?
Yeah, SDS page, it was there, but SDS page, what happened? As I told, yield in crude form, it will be very less. Okay, it is secretory.
Yeah,
So we'll take the secretory portion. It won't have any other, not major any other protein, but we run the gel. In normal crude yeast, if we run, what will happen is like you will get many protein, many bands and everything. Here we'll get only our protein as main band, but it is not so robust. So what we'll do, we'll increase the volume of culture first. Okay, so we will have culture like 1 liter. From there, what we'll do little bit, we'll centrifuge and remove the yeast. Only media is there. That media will add will protein precipitating agents, so that will concentrate the protein. So we have a little bit concentrated protein with us. So that protein we done for this purpose actually. So if we do normal as such from media and do Western, not Western, like normal SDS page, we will see a faint band. So it is not suitable for showing. That is why I told episomal expression, you will have like a better band in normal SDS page. So no need to bother. In the equal and all, you will have a really big thick band, normal SDS page. But in yeast normal case, you won't get a thick band. In Saccharomyces episomal expression also you won't get. So Western is the way to get detect it. Otherwise you will feel like your protein is not getting expressed.
Yes,
So
yes.
SDS page many times what will happen, it will become like a false negative. So it is positive, it is getting expressed, but you won't have that kind of equal kind of expression in yeast.
Okay, please continue
Okay, so this,
quickly.
so I'll just conclude what I am telling. So the steps in recombinant protein expression yeast. So we have to go for an host organism based on the protein only. So we have to see our protein, go through the sequence thoroughly, see what else are there in that one, what are the glycosylation or whatever is happening. Then only we have to select. Next step, as I told you already, expression is always limited. Even in we go for episomal, which is having multiple copy number of gene, we have to go for codon optimization so that our yield will increase. Next is always better to go for a secretory signal, especially we want to work with little bit with the protein. So it enables us like purification. Okay, otherwise what will happen, getting out protein from a system is like really difficult, and it degrades. Even if it don't degrade, we start using some agents to break apart the protein. What will happen, it slowly affect the functions also, function of the protein also. folding and everything there, that also will get affected, and purification, everything it will get affected. So most of the people, I think like unless they are going go for like very small scale production and function analysis, most of them opt for adding on secretory signal. So last point is integrative expression. It is stable and it is hence it is cheaper. So if we want to work long term with one protein or something, or we want to scale up or industrial or this one is there in application is there in mind. So always go for an integrative expression because that is the only way it can be taken to higher scale. Okay. So I thank Dr. Gopal to give this opportunity. And
अरे
I have a small lab at IACB. So these are my PhD students, Sunita Das, Anjali, Amrita Gonda, and Ananya Kumar Ghosh. So I'm funded by CSIR and I have a startup research grant from NRF. So thank you. So now I'll take questions.
Yes, thank you very much. It's good